Hello everyone. So I'm going to discuss about Unit 8, which is Biology in Human Welfare. I'm going to take a very small topic in this chapter, which is AIDS. So let's dive into the video. AIDS. So what is AIDS? AIDS is nothing but acquired immunodeficiency syndrome. So by the name itself, we can actually understand that it means the deficiency of the immune system. Here the immune system becomes weak, and syndrome here suggests the group of symptoms. So when a person actually has AIDS, that person will be exposed to a variety of diseases, even common diseases, and they will likely to get those diseases because of the weakness in their immune system. So cause of AIDS. AIDS is caused by the human immunodeficiency virus. Let's look into the structure of the virus. So it is round in structure and it has glycoprotein spikes on its surface. These spikes will only bind to the CD4 receptor. So it has a capsid here, and this is a capsid, and then there are two RNA genome, single-stranded RNA, and they have some viral proteins and some enzymes called as reverse transcriptase. So there are three enzymes involved in the life cycle of HIV. First is reverse transcriptase, second is integrase, third is protease. So how does the HIV get transmitted? It gets transmitted because it spreads only through body fluids. So because of that, it gets transmitted through sexual contact, sharing of infected needles, especially in the case of intravenous drug abusers, and by transfusion of contaminated blood and blood products like from blood banks, and from infected mother to her child through the placenta. Who are at high risk? Those who have multiple sexual partners, drug addicts who take intravenous drugs. I mean, those intravenous means the drugs that are taken via injection through the veins, and individuals who require repeated blood transfusion, for example, certain diseases such as anemia and kidney diseases and cancer, and children who are born to HIV infected mothers. So they are at high risk. So let's look into the life cycle of HIV, which is very important. So this is the structure of the HIV which I discussed here, and these are the glycoprotein spikes here, and this will only bind to the CD4 receptor on the cells. So what are the cells that have CD4 receptors? So here I told you about glycoprotein. This is the HIV virus which has glycoprotein on its surface, and this will actually bind to a cell which has a CD4 receptor. So example, this is a CD4 receptor. So these cells that have CD4 receptors are the ones are actually T cells, macrophages, and monocytes. This particular cell is either a macrophage, T cell, or a monocyte. So this HIV binds to CD4 receptor, and then what happens? This is the first step: fusion of HIV to the host cell surface. Second step is when HIV RNA reverse transcriptase integrase and other viral proteins enter the host cell. So this particular envelope that you're seeing right now, this particular envelope is this envelope dies down. It doesn't get incorporated into the cell. It only only the viral genome gets incorporated into the cell. After that, the viral genome. Now you need to understand that the viral genome in the HIV virus is actually in RNA form, and the viral DNA and the genome in the infected cell is actually in the form of a DNA. So how do we incorporate RNA into the DNA? So the RNA needs to be converted into DNA. This is done with the help of an enzyme called as reverse transcriptase. So I'll talk about this enzyme. So we are aware about DNA getting converted to RNA and RNA getting converted to protein. DNA converting to RNA is actually transcription. RNA getting converted to protein is translation. And RNA getting converted to DNA is reverse transcription. This is done with the help of an enzyme called as reverse transcriptase. Transcriptase. For RNA to get converted into DNA, it needs an enzyme called as reverse transcriptase. The process is referred to as reverse transcription. So here, the RNA genome has to be converted to DNA. Now it has become the viral DNA with the help of an enzyme called as reverse transcriptase, and then there is a enzyme called as integrase which helps in the integration of viral DNA into the host cell DNA. The host cell DNA here is in blue color and the viral DNA here is in the yellow color. So it gets incorporated with the help of an enzyme called as integrase. I mentioned three enzymes. One is reverse transcriptase, integrase, and protease. Then this viral DNA starts transcribing to RNA. So it gets transcribed to genomic RNA and mRNA. This mRNA then gets translated into proteins. And then this genomic RNA with the viral proteins, they go to the other end of the cell surface. When they reach the other end of the cell surface, they start to bud off. And then when they start to bud off, a viral protease is actually used, which cleaves the new polyproteins to create mature infectious virus. This is the process and life cycle of HIV. Let's revise. So first, the HIV virus is glycoproteins. With its glycoproteins bind to the CD4 receptors of either T cells, macrophages, and monocytes. And then what happens is only the viral genome gets into the cell, and then envelope does not get into the cell. And then the viral genome, in order to get integrated into the host cell genome, it has to be transcribed to DNA. And when it does that, it uses an enzyme called as reverse transcriptase. And then an integrase enzyme is actually used, which helps in integration of the viral DNA into the host cell DNA. The host cell DNA starts the process of transcription, and we have genomic RNA and mRNA. The mRNA gets translated into proteins, and the genomic RNA along with the mRNA, they move to the side of to the end of the cell surface, and they start to bud off. And then the protease is actually used to cleave the polyproteins to create a mature HIV, and this actually infects other cells. This is the life cycle of HIV. This is the text that I have from which I have actually explained. Now we'll look into the life cycle of HIV according to the CBSE textbook. As I was going through the life cycle of HIV and CBSE textbook, it was not apt. The information was not totally detailed. So in order to understand it, I gave a detail. I went through the papers and then I understood the concept. So let's look into the textbook. The virus enters the macrophage after entering the body of the person. So here it can enter a macrophage or a T cell or a monocyte. RNA genome gets replicated to form viral DNA by enzyme reverse transcriptase. I have mentioned that RNA gets reverse transcribed to DNA with the help of an enzyme called as reverse transcriptase. Viral DNA gets incorporated into the host cell's DNA with the help of an enzyme called as integrase. And directs the infected cells to produce new viruses. Macrophages continue to produce virus particles, thus acting as a HIV factory. So this is like, this cell here is referred to as an HIV factory because it continuously produces HIV virus. At the same time, the virus particles enter into helper T lymphocytes, so it infects the helper T cells. The number of T cells actually decreases. So during this period, what really happens is fever, diarrhea, and weight loss really happens. And you need to understand that T cells actually are required for immunity. It actually helps build up the immunity. So if the number of T cell decreases, the immunity also decreases, and as a result, the affected individual will have a very weak immune system and will not be able to have an immune response against common diseases and common bacteria like mycobacterium, parasites like toxoplasmovirus and fungi. So they're exposed to common diseases such as common cold, typhoid, pneumonia, and all of that. Diagnosis of AIDS. Diagnosis of AIDS is actually done by ELISA, which is enzyme-linked immunosorbent assay. Treatment of AIDS. So basically, it has no cure. We actually use antiretroviral drugs, but this can only prolong the life of the patient, but it cannot prevent the death. So death is inevitable in this case. So the last slide, prevention of AIDS. So awareness and precautions are actually taken up, and they've been started since 1991. And there are some main strategies of prevention here. First is making the blood and blood banks safe from HIV, and then we need to use disposable needles and syringes, and free distribution of condoms, and advocating safe sex, and controlling drug abuse, and promoting regular checkups for HIV and susceptible people. So all of these are actually some main strategies to prevent HIV. Thank you for listening, and thank you for watching. So I'll be coming out with many more videos, and please share and subscribe to my channel.